Welcome to the last topic of the course where we investigate genetics, which is largely due to Gregory Mendel, who was over a couple hundred years ago the founder of Mendelian genetics. Gregory Mendel had used pea plants due to the fact that they had many identifying characteristics that could be identified and passed on to the next generation. For instance, he was looking at seed shape, which was having either round or wrinkled characteristics. Also, he noted differences of seed color that could be of either yellow or green traits. He also was looking at flower colors, which these pea plants were either purple or white flowers, and even seed shape and seed color, where the seed shapes could be identified as inflated or contracted, or that of the seed color, the fruit color, would be yellow or green. He was also looking if the flower position was either at the axil position or the terminal position, and then he was also looking at the height of the plants, that some plants were growing either tall or others were being short. And due to this, he was able to control the pollination to where he could regulate what plants were being crossed with others to determine the inheritance probabilities of the next generation. And because of Mendel's investigations, he is known as the father of heredity, which is how alleles, which are the alternate forms of the genes, are being passed along and inherited from one generation to the next generation. As we see here with this family, how traits are being passed along, such as hair color. And these traits are each found on the homologous chromosome pairs. So as we'll see, that there are many different types of genes. Roughly in humans, we have 23,000 genes scattered throughout our 23 pairs of chromosomes, which means that we have on average about 1,000 genes per chromosome. But because of the fact that we receive our homologous chromosomes during the fertilization process between the sperm and the egg cell, that they don't necessarily have the same exact information found at the gene positions between the homologous chromosomes. And as we will see, that we can start to use letters to represent the different forms of the genes, which are known as alleles. However, in a gene sequence, we have the same genes at the same region of the homologous chromosome, but they can have the alternate forms of those genes known as the alleles that are represented as the information that is passed on to the next generation. So for most genes, there are always two forms of the alleles that are found at that locus or the position of the chromosome that carries the gene. Now here we can see that the gene for finger length could have either the dominant characteristic, which would be represented by the uppercase letter, or the recessive characteristic, which is represented by the lowercase letter. So to this characteristic, when we see that we have one dominant and one recessive, that the dominant trait is going to be expressed and the recessive trait is going to be masked. So when we have one of each, that this individual would have the dominant short fingers. So in viewing the possible combinations of alleles, that there are some terms that we can describe as the genotype, which is the combination of alleles at the gene, where there could be two dominant alleles represented by the two uppercase letters, and this is referred to as the homozygous dominant genotype, as this homo refers to the same, and then the zygous is the zygote that is the first cell to receive the pairs of homologous chromosomes. So when the two together are dominant, of course, then it would be termed homozygous dominant, but there could also be the possibility of two recessive alleles together, so this would be then the same similar term homozygous, but it is then specified as recessive. We also could have the possible combinations of one dominant and one recessive, which then we need to use the alternative term heterozygous, where hetero refers to the different, and so by having one of each being different is heterozygous. We can also see, though, that we can see from the genotype what the expression should be based on the allelic combinations. If there is a dominant trait, that we would then be seeing the dominant expressed as short fingers, and this is the phenotype. So the phenotype is the physical expression. If it is heterozygous genotypically, then it is still having short fingers because of the fact that the dominant is expressed and the recessive is masked. But the only way to get the recessive trait being expressed is having the two recessive alleles together in the homozygous recessive form, which phenotypically would be known as long fingers. So as you can clearly see, heterozygotes are just allele uneven. As Mendel was looking at pea plants crossing traits going from one generation to the next, he noticed that if he took a purple flower, which was a true breeding population, meaning that they were homozygous, and he crossed it with a white flower, which was also a true breeding population being homozygous, that he then produced what is known from the parent cross, the F1 generation or filial 1 generation, which had all of the same characteristics, because this then showed that the purple flower was the dominant trait over that of the white flower, to where all of the F1 generation was heterozygous genotypically and also purple phenotypically. So then, with this knowledge, if he had on the homologous chromosomes one purple allele and one white allele being heterozygous, that they were all, of course, purple, but then knowing how these traits then are passed along into the next generation, we must follow these homologous chromosomes through the process of meiosis to identify what alleles are possible into the gametes. So, in the interphase stage of the cell cycle, that the S phase of interphase will have the homologous chromosomes replicate, so that now they have the sister chromatids being held together at the centromere by cohesin proteins. So if it is that they replicate, we have the two sister chromatids here with the dominant allele, and then the two sister chromatids here with the recessive alleles. And through meiosis 1, that the homologous chromosome pairs will separate into the two daughter cells. And then continuing on to meiosis 2, in meiosis 2 is where we separate then the sister chromatids into the 4 haploid daughter cells that can eventually mature into the gametes. But if we notice here that these two gametes will have the same trait, being the dominant allele, and then these two gametes here have the recessive trait as the white flower, and the dominants were that of the purple. And this is what we then set up on the Punnett square to show how these alleles could be passed on into the next generation. So here we see two heterozygous purple parents that would be able to be crossed, where half of the gametes would contain the dominant allele, half the gametes would contain the recessive allele, and this would be the same for both parents. So that here where we cross, we use the Punnett square to place this allele with this allele, pairing the gametes in this possible fertilization event producing the zygote, which would be homozygous dominant. But then we also pass this one down and this one across, which could then produce a heterozygous individual. But then this gamete could have also fused with the alternate gamete here, making another heterozygous. And these all three would be expressing the purple flower color phenotypically. And the only way to get the other phenotype, which is the white flower, is to have this recessive allele coming across and pairing with this recessive allele, which would make the homozygous recessive and express itself as white flowers. And this gives us a 3-to-1 ratio. So let us look here at a genetics question that is looking at the same pea plants that Mendel was looking at hundreds of years ago, where yellow seeds are dominant to green seeds. Now we notice that we use the same letters for the same gene to pair those together, and we have the uppercase letter to represent the dominant yellow allele and the lowercase y to represent the recessive green allele. So if we had a genetic cross of two plants that were both heterozygous for the seed color trait, here we can then follow through steps to find how the fraction of the offspring would have expressing yellow seeds. And this is known as a monohybrid cross, which is a single factor cross, which is looking at one trait being passed along into the next generation. So here we will follow these 5 basic steps to identify how to perform the Punnett squares. First, we will assign the symbol for each allele. Here we have the dominant allele is the yellow seeds, and the recessive allele is the green seeds. The next step would then to be determining the genotypes of each parent, which in the question it was stated that both of them are heterozygous, which has one dominant and one recessive for each of the parents, which are genotypically heterozygous but phenotypically yellow seeds. We will then determine all the possible gametes by taking those alleles and separating them into the possible haploid cells, and then we put those on the Punnett square that then can be used to determine all the possible combinations of the offspring. Then lastly, we will then determine the phenotypes of each offspring from their genotypes in the Punnett square. Here we can follow this through the monohybrid cross, starting with the P generation, where we had our true breeding population, where we had the yellow seeds, which were homozygous dominant, and the green seeds that were homozygous recessive. And in the gametes that they can produce, the homozygous dominant only has the dominant allele, the homozygous recessive only has the recessive allele, and when paired together in the F1 generation, that this explains why 100% are going to be expressed as the yellow phenotype, due to the fact that they are all going to be heterozygous. But then when we pair the heterozygous plants together and then produce the F2 or filial 2 generation, that this then makes it to where we have this characteristic 3-to-1 ratio again, where we have the dominant alleles pairing together in this one, which is homozygous dominant, the dominant and the recessive occurring twice between the pairs of alleles, being 2 that are heterozygous, which are all expressed as yellow seeds, and then the last one, which is the only one that has the recessive alleles, being homozygous recessive, and can express the recessive phenotype of green seed color. So let's step through this process with a similar cross of still looking at the seed color. Here we'll have our yellow seeds still dominating the green seeds, but if the genetic crosses of 2 plants, one that's heterozygous for the seed color trait and another with green seeds, we will see that the fraction of the offspring will have changed from the previous question. We first identified our alleles, the dominant being the yellow, the recessive being the green. We identify the genotypes of the parents, where one was described as being heterozygous and the other was green, which can only be expressed in the homozygous recessive genotype. We then follow the alleles into the gametes, where the heterozygous individual can make gametes with either the dominant allele or gametes with the recessive allele, but we note that these are haploid because they only have one of the alleles of the homologous chromosome pair, and the other parent, being homozygous recessive, could only make gametes with those 2 recessive alleles. After the gametes had then been set up on the sides of the Punnett square, you just put those across and then put those alleles down to pair them together to form the zygotes. Once then you identify all of the genotypes of the new generation, we will then be able to determine their phenotypes. Here we notice these 2 are going to be heterozygous, expressed as yellow seed color, and these 2 are homozygous recessive, being expressed as green seed color. So it is in a 1-to-1 ratio, or 50% yellow, 50% green. Not all genes are known to be dominant and recessive, but some are described as being incomplete in their dominance. And this is where you're going to notice that the way in which the alleles are paired together will have unique phenotypes. Here we'll see that we use the C letter to represent the allele, and then this superscript R is the entire allele that would be having the red flower color produced. And so if you have 2 red alleles together, of course it would be a red flower. However, we notice here that the way that the white alleles are represented, we still use the same letter C to tie it to the gene, but this W and the R does not show that one is dominant over the other, and when you have the two white alleles together, this of course then would produce a white flower. Now what is unique is that when you had a dominant and a recessive allele, that the heterozygous form was only expressing the dominant characteristic. But here when we have the heterozygous, where you have the one red allele and one white allele, that in incomplete dominance, that it has a blending of the characteristics which produces a pink flower. But note, there are no pink alleles, it's just the way in which these alleles come together and are the combination of those traits being expressed. So that every unique phenotype has a unique genotype. So let's look at this if we were to cross two pink flowers, which because it is pink, you know that the alleles would have to have one red allele and one white allele genotypically for the specific phenotype. So we see here that we have our red and our white alleles for each of the parents being pink. And these then, we follow the same rules that we separate these alleles into the gametes, where each parent can make a red gamete or a white gamete carrying a cell, and so when they come together that these two produce red flowers 25% of the time, but we note that these two combinations here and here are now heterozygous, so just like the parents are expressing pink flowers, and this is 50% of the time, and then the last combination that we see are the two white alleles coming together, which 25% of the time make white flowered offspring. There are also those differences between the sex chromosomes. So those that are found on the sex chromosomes are referred to as sex-linked traits. And any gene that is tied together on the same chromosome is considered linked. However, when we look at the X chromosome and the Y chromosome, that male combinations have one of each, whereas females have two X chromosomes. But we also note that the Y chromosome contains fewer genes than the X chromosome, that some of those are vital to the male expression, but because of this, there are genes that are located on the X that are not located on the Y. So that in a male, you are going to have traits to be expressed that are on the X chromosome without an alternative to be paired on the Y. So for instance, if you are a male with a recessive allele on the X chromosome, if there is no alternative on the Y, that is going to be automatically expressed within the males. However, being a female with the two X chromosomes, that a female could have one of her X's with the recessive allele, but she's not necessarily going to express that because her other X chromosome could have the dominant allele, and in this situation, if she has the heterozygous pairings, she is considered a carrier because she has a recessive that could be expressed into the next generation that she carries, but she herself does not express the recessive trait. And so due to the sex chromosome combinations, we will note that in crosses that males have a higher tendency of expressing recessive sex-linked traits because all they require is that their one X chromosome carries with it the recessive allele, whereas in females that they require both of those to be recessive in order to express it, which is going to be reduced in the frequency of the expressions. So a common recessive sex-linked trait is that of color blindness. So if we look at here a colorblind male having a child with a female who is a carrier for color blindness, meaning that she is heterozygous, we could then still follow through with the same procedures to identify what probability the children have of being colorblind. Here we see the male that on his X chromosome for being colorblind carries with it the recessive allele, and then nothing on the Y. And here we have our female, which was described as being heterozygous, so one of her X's has the dominant, the other X has the recessive. So now we just then separate those alleles, seeing that the female could produce gametes with either the dominant or the recessive X, and then the male can produce gametes with his X chromosome that has the recessive or the Y chromosome. And then as we look at the pairings through to the offspring, we will notice that this female is going to be a carrier. This female actually expresses color blindness, and then the male, which receives his X chromosome from the mother, has the dominant allele 50% of the time of the males that would then have normal vision, but then here the other male would be having the recessive allele, which would be a colorblind phenotype. When looking at blood types, we find that these traits are being passed along by more than just two possible alleles, which this is an example of multiple alleles, as well as a characteristic known as codominance. As we'll see here with the alleles, that there are A alleles that are dominant as they are written with this immunoglobulin protein that is produced, which is why they use the letter I generally, and then this uppercase A to express the dominant A allele. But if we notice that B alleles also are dominate, and we'll have the I with the uppercase B, but when looking at the O allele, it is written as the lowercase, which is why it is recessive. So A and B both dominate over the O allele, but this is a little bit different of how these are expressed. The A and B alleles, when together, form AB blood because they both produce these proteins that are found on the red blood cells in the form of the A and the B, which is not a blending of characteristics like incomplete dominance, but are actually both equally expressed in the AB blood. And the only way to express O blood, because it is recessive, is to have the two O alleles, which generally is not going to be expressed at all because there is no immunoglobulin protein. So this here in itself is not producing any protein, which is why it's a universal donor and will be able to be passed along to everybody. But they themselves can only receive O blood, which, when we start describing the blood types, are their phenotypes. So here we see O phenotype expressed when the genotype is the homozygous recessive, AB blood type, which is only produced with the A and the B alleles coming together, and then B could actually be from two different genotypes. You could have the two B alleles together, or one B allele and one O allele, but because B dominates O, it is still only B blood. And the same for A blood, that you could have the two A alleles together, or the A and the O genotype, and this still is only expressing A blood type. So if we were to view these codominant crosses where we had two individuals that were A and B and another individual that was A and B, that these could pair together forming A blood 25% of the time. These could come together forming AB blood 50% of the time, and then the two B alleles could come together forming B blood the other 25% of the time, which here gives us this genotypical ratio of a 1 to 2 to 1 ratio of A, AB, and B blood, and there would be a 0% chance of having O blood type. So the previous examples were looking at monohybrid crosses having one trait being passed along into the next generation. Now we will look at a dihybrid cross, which is di referring to the two characteristics being passed through. So when we view here in pea plants again, we will look at the yellow and the smooth, which are the dominant alleles, and then we're going to see how they pair with the recessive alleles of green and dented seeds. So if it were that we have our parents in this true breeding population, this one being homozygous dominant for both traits, this one being homozygous recessive for both traits, that they will always produce the same types of gametes, where if you're homozygous dominant, the dominant for smooth seed shape is going to be passed along, as well as the dominant for the yellow seed color into the gametes. But we notice that the gamete being haploid has only one of each of those alleles. But the same would go for the homozygous recessive, but this would only then be carrying those recessive alleles. The recessive for the seed shape, which would be dented, and then that of the seed color, which would be green. So in the F1 generation, that we would see that they pair together again, producing heterozygous individuals 100% of the time. And these would then be expressing the dominant trait of smooth and yellow characteristics. But remember that these genes could be found on different chromosomes, and then there's also events that take place in meiosis, such as crossing over, that can increase the variation to the gametes, as well as if they're on different chromosomes, the independent assortment will increase the variation. So if we note here that we have dominant alleles on the blue chromosome, recessive alleles on the purple chromosome, that in the events of crossing over, that now you can exchange them, and this forms recombinants when crossing over takes place that has the now two different alleles that were not found on the original chromosomes that have a dominant and then the recessive Bs, or the recessive A and the dominant Bs, which are now new combinations of gametes that must be taken into account to see how these gametes could pair together. But remember also, independent assortment increases the variation by having the homologous pairs of chromosomes align independently, which then could make alternative gametes. So when these alleles separate, they will be paired together with the different alleles from the other genes as possibilities. So if you could remember this technique, which is known as foiling, where you just would identify all the possible gamete combinations, taking the dominant S and pair it with the dominant Y, and we see that in the gamete here, but then you just pair the dominant S with the recessive Y, and we see that gamete is this possibility, and then do the same with the alternate allele for this gene. It could pair with the dominant Y, and we see that in this combination, and then the last combination is taking the recessive S, pairing it with the recessive Y, and these are the four possible gametes. And this requires that we place these along an extended Punnett square to show the possible gamete types of the parent of the F1s that were originally heterozygous for both traits, and we would do that for the same other parent. And we place the alternative parents' gametes along the other side. And then following along with the same steps, we then cross the possible gametes. So these gametes are going to be filled in going down the columns, and then we take the alternative gametes and put those across the rows. And if you notice, we put the letters back together to help to simplify identifying those characteristics of how the genotypes are pairing up. And when now we look to identify the phenotypes, anything that has at least one dominant allele for both genes is going to be expressed as smooth and yellow. The genotypes that have at least the dominant S allele and the recessives of the Ys are going to be smooth seed shape, but because they're homozygous recessive for the other gene, are expressing the recessive green phenotype. These here are now homozygous recessive for the S alleles, and at least having one dominant Y, so they express the recessive dented seed shape, but the dominant yellow color, and only one is going to be homozygous recessive for both of the characteristics, being dented seeds with green seed color. So in the F2 generation, we get this characteristic 9:3:3:1 ratio of the phenotypes. But the only difference here in our dihybrid cross is that when we make that parent generation into the F1, which is genotypically heterozygous for both of these traits, you just have to remember that the gametes are going to have this increased possible variation that then, following through the same steps, come together in giving us the 9:3:3:1 ratio. And you should feel good about yourself if you have reached this slide, because that's all of the information for the course. And good luck with the last examinations and finals, people.